Welcome to an overview of the gene editing revolution, framing strategic insights and future frontiers for enterprise and innovation. This slide introduces therapeutic advancements, agricultural applications, bioethics and policy considerations, and the shifting investment landscape, setting the stage for how these technologies create market opportunities while reshaping research, commercialization, and long-term strategic planning. This slide traces the evolution from early protein-guided systems like zinc finger nucleases and TALENs to RNA-guided CRISPR-Cas systems, highlighting how complex protein engineering gave way to simple base pairing for targeting. The transition dramatically lowered barriers, reduced costs and time, and expanded global accessibility, enabling many more labs and companies to translate ideas into practical gene editing solutions. Here, we walk through the CRISPR-Cas9 precision editing workflow, from guide RNA binding to Cas9-mediated double-strand breaks, and the cell's repair choices. The diagram shows non-homologous end joining producing indels and gene disruption, or homology-directed repair, using a template for precise correction, underscoring how targeted cleavage and cellular repair pathways enable rapid, precise genetic modifications. This slide introduces next-generation editing with base editors and prime editors, which move beyond simple cuts to enable single nucleotide conversions and sequence insertion or replacement without frequent double-strand breaks. Base editors chemically convert bases for point mutations, while prime editors write short new sequences, together expanding correctable mutations and broadening therapeutic and agricultural possibilities with greater precision and fewer unintended outcomes. Ex vivo cell-based therapies follow a clear, four-step path: patient cell collection, targeted ex vivo editing and engineering, expansion with quality control, and reinfusion followed by monitoring. The slide illustrates how this approach is used for sickle cell disease, beta thalassemia, and CAR-T cancer therapies, emphasizing durable clinical benefits, rigorous manufacturing standards, and the regulatory milestones that underpin safe, effective treatments. In vivo delivery mechanisms rely on lipid nanoparticles and adeno-associated viral vectors to ferry editing machinery directly to tissues after systemic injection, with clear examples targeting the liver and the retina. This slide highlights the therapeutic advantages of minimizing complex cell manipulation, eliminating some transplantation procedures, and enabling in situ correction of genetic defects for diseases that require direct tissue delivery. CRISPR is accelerating agricultural innovation by precisely modifying native genes to enhance traits like drought tolerance, disease resistance, and nutritional value without introducing foreign DNA. The slide explains how gene-edited crops can bypass some transgenic regulations in key markets, shortening breeding cycles from decades to months and enabling faster market access and more rapid realization of trait improvements for global food systems. This slide presents agricultural case studies: hornless dairy cattle, PRRS-resistant pigs, and fast-growing salmon, to illustrate welfare, efficiency, and market impacts. Each example shows how targeted edits can improve animal welfare, stabilize supply chains, or boost productivity, and the synthesis highlights that economic viability and consumer acceptance hinge on traits that mirror conventional breeding outcomes while delivering clear societal benefits. Unintended genomic edits present off-target risks that could disrupt important regions and increase tumor risk, and this slide outlines mitigation strategies. Sensitive detection methods and engineered high-fidelity enzymes reduce off-target activity, while preclinical screening, in vitro validation, and in vivo safety models form the clinical imperative to minimize risk profiles, recognizing that rigorous assessment is essential, though risks cannot be entirely eliminated. Heritable germline editing raises complex ethical and policy concerns spanning consent for future generations, the boundary between therapy and enhancement, potential social inequalities from unequal access, and unforeseen ecological impacts. The slide emphasizes international responses calling for moratoria, stricter oversight, and coordinated governance frameworks to manage intergenerational effects and to ensure ethical, equitable decision-making at a global scale. Emerging epigenetic tools offer reversible gene regulation by modifying DNA methylation and histone marks without changing the underlying sequence, preserving genome integrity while altering expression. The slide highlights therapeutic applications in diabetes, pain management, and neurodegeneration, and notes advantages such as tunable therapeutic windows and precision control, presenting epigenetic interventions as flexible, potentially reversible complements to permanent edits. Democratization and global governance balance broader innovation access with biosafety and biosecurity responsibilities. By leveraging enabling factors like falling costs, open-source protocols, and cloud lab services. The slide connects these enablers to local impacts in developing nations for health and agricultural challenges, and argues for harmonized regulations, ethical standards, and equitable oversight to ensure innovation is paired with safe, fair global governance.